My name is Atul, and today I am going to talk about this interesting problem of measuring gene expression using high throughput technology called microarrays. So, what is gene expression? What are genes? Right? So, recent advances in technology have completely transformed biology. Right? We all know the central dogma of biology, which says human body contains genes. Genes are essentially small fragments of DNA which is coiled in a double helix inside our cells. These genes are responsible for encoding proteins, so they result in proteins being created. And these proteins in turn control the function of our body. So genes are the fundamental building blocks of life. Now, the problem is these genes do not act in isolation. It's the interaction of multiple genes together which results in a particular behavior of an organism. And hence, if you want to understand how genes impact function, we need to study thousands of genes together. And what we need is a high throughput and a scalable technology which will let us observe all these genes together or simultaneously. It will be too painstaking to go one at a time because there are hundreds and millions of genes. So what is gene expression? Think of it as a gene being active or manifested. When a gene is expressed, it really does the job it is supposed to do, which is to synthesize a functional product like protein or mRNA or whatever it is supposed to do. And the pattern of gene expression, which genes are active, characterizes the current state of a cell, which essentially characterizes the state of the organism of which the cells are part of. So I'd like you to look at a quick video which explains what gene expression is. Wonderful animation, and I hope this is a good idea.
Gene expression is complex. To start transcription, several proteins are often required to bind regulatory elements on the DNA. This step in the process is where DNA is read and a complementary strand of RNA is produced. The RNA has to undergo many changes, including the systematic removal of RNA sections not needed to code for the desired proteins. RNA then takes the information encoded in the DNA out of the nucleus. In a process called translation, ribosomes interact with the messenger RNA and transfer RNAs to create the chain of amino acids coded for on the DNA. This amino acid chain undergoes changes in configuration, including specific folding. Each protein takes on a 3D shape required for a functional end product. As the new protein leaves the cell and takes on its role in the body, the cycles of cell function and gene expression carry on. The amazing symphony of cellular interactions continues, and the music, which is life, plays on.
So coming back, right? So what is a microarray? Right? It is a device which helps us measure the activity or expression level of thousands of genes simultaneously. And what are the applications of such a technology? Well, numerous, right? Identification of complex genetic diseases. What gene or a set of genes are causing a disease condition? And you do that by doing comparative studies between a diseased tissue and a normal tissue. What's different in these two tissues? What caused cancer, and so on. It's also useful in drug discovery. When you apply a drug, the state of the cell changes. So some genes get expressed, some don't. Now what changed? As a result of which, what function altered, and so on. So it's an extremely useful technology. How does a microarray work? So you take a substrate like a glass or maybe nylon, and you have thousands of spots on that. Each spot contains a small fragment of DNA which uniquely identifies the gene. This is called a probe. Then you take your diseased tissue, normal tissue, you dye the normal tissue with green and the disease with red, and you wash this fluorescently labeled sample onto the microarray. Complementary strands stick, and so they bind to the spots, and what you do is measure the amount of genes fragments accumulated in each spot. And that can be done by measuring the intensity of red light or green light fluorescence at each spot in the, on the microarray. So this is a typical fluorescence image generated out of a microarray. You notice there are lots of spots here arranged in a rectangular fashion. Some are red, which means the genes in the diseased sample or your experimental or treatment sample are highly expressed. Some are green, which means the genes in the normal sample are highly expressed here. Yellow means both are expressed and black means none are expressed. So by using this, you can now quickly tell that what is different in a disease sample as compared to a normal sample. And for these many genes, thousands of genes, not one at a time. So this fluorescence image is the starting point for all the analysis you will do subsequently with this microarray. So you start with this data, then you apply some numerical analysis, some machine learning, finally tie it back to biological inference, and make some meaningful conclusion, right? So once you've extracted all your measurements from a microarray image, what you get is a square grid. Each row represents gene 1, gene 2, each row represents a gene, and so on, right? And the column represent the states, right? This is a normal sample. This is a disease sample at 5 seconds or 5 minutes. This is the disease sample at 10 minutes or 10 hours, whatever the measurement might be, and so on. So you are essentially tracking the activity level of a gene across time. It need not be time, it could be multiple experiments as well. So you have n such columns, and you're dealing with n dimensional points. And what you are working with is, if you have m such rows, then you have n dimensional points, and you are trying to now work with numerical analysis for this data. So let's look at the analysis you would do. You first filter the results to remove errors, outliers, experimental errors. You would normalize this data. Then this is an important step. You will analyze which genes are truly differentially expressed and not as a result of experimental errors. And you would use various statistical techniques to do that, and I'll talk about that later. And finally, apply some clustering and machine learning techniques to make sense of this data. So what kind of statistical techniques would you use, right? Use techniques like t-test or analysis of variance, which will tell you whether your control group or treatment group are truly differentially expressed, right? So you would run this test and remove data which does not pass this filtering criteria. And you will be then left with a smaller set of genes, smaller set of rows in your 2D table, which you will work with later. Now, once you have a statistically clean data, you can ask questions like, give me genes which are similar, which react similarly in this disease tissue or to a drug treatment or whatever it might be. So k-means clustering is a very popular technique, one of the popular techniques here. What you do is you group the genes into k distinct groups or clusters, and each cluster has similarly behaving genes. So in this image, for example, this pink cluster is one cluster where the genes are similar. They are at the similar locations in the n-dimensional space. Similarly, this green cluster is somewhat similar. And then you might further study the aggregate properties of this cluster or the genes inside that cluster, right? If the genes are similar, it's very likely they are participating in a similar function or they are part of a similar pathway and so on. Hierarchical clustering is another popular technique used for grouping genes together. So what you see here is these set of genes are similar in some way, right? They are expressed more at 4 hour, 6 hour, 8 hour, 12 hour intervals, and then again in somewhere in the control group. So what this technique does is brings similar genes together and dissimilar genes far away. Similarly, these set of genes are similar. So now you would do further studies on these similar set of genes. But clustering is not enough. You need to go beyond that and use some better techniques to infer more about this data. And this is where machine learning comes in. So classification techniques. So one of the often used supervised machine learning techniques is decision trees. What it gives you is an empirical set of rules which will help you classify data into the categories you have identified for the for the genes. So it will give you a rule that if this level is less than 1 and if CLN1 is greater than 0.8, then it means that this set of gene is upregulated, and so on. And this data is inferred from your data set with some human input. One of the most popular techniques in recent time is support vector machines, right? The benefits are it's unsupervised, doesn't need any user input, and it finds an optimal solution rather than a greedy solution. What it amounts to is if your results are into two categories, then it will find the reason or the separation between the two categories. And the way it does that is projects the data into higher dimensional space and finds a separating hyperplane between this data. And the equation of that hyperplane or the characteristic of that hyperplane might give you the reason because of which you have a clean separation between your diseased tissue and a normal tissue. And then you can analyze that further. So this is about numerical analysis, but this is not enough. You have to go beyond. So you need to now take this numerical data and tie it back to biology to make sense of it. So you would first annotate the genes to get functional information. Algorithms like BLAST are extremely popular here. You would use text mining, query the existing literature which is all available online to find are the genes having similar function? Are they part of the same pathway? If they are part of the same pathway, then very likely they affect each other, either positively or negative correlation. Are they referred together in literature? So text mining and these kind of techniques are employed. So let me share you some places where you can get more information about this. So let's go to Wikipedia and search for DNA microarray. And this is a good starting point where you can get a very basic high-level introduction to what microarrays are, which companies produce these devices, what are the general scientific principles of an experiment. Here you see popular gene chips from a company called Affymetrix, which was a pioneer in this field in the mid-90s and late 90s. Different types of arrays and experiments and devices and so on. You can find more information on the Affymetrix website. Right? So if you go to the site and say search for microarray, it might take you to some pages where there's basic information about the products of this company. So you can go through it and get a good idea about what kind of instruments are available, what kind of solutions are available, how do they work, and so on. It's a highly sophisticated field there. Finally, you can go to online databases which I referred to. So PubMed is one of the very popular databases, and all kinds of biological literature is all available here, and you can search for them, right? So you can let me, for example, search for PubMed, and I want to say, give me information about hemoglobin, right? And it returns me 146,000 results, huge number of results, with all kinds of information about where this protein was referred to and what kind, and lot more data which you can use for your text mining information. So I hope I've been able to convey a very brief introduction about this technique and gotten you interested. Thank you.